Good morning, everyone. Today we are going to discuss about a topic called Meselson stahl experiment. Meselson stahl experiment. Stahl experiment. Okay. Meselson stahl experiment. Do you know what is the reason of this experiment? This experiment is conducted to know the mode of replication. How does the DNA replicate? What is the mode of replication? This experiment was conducted by Meselson stahl in 1958. 1958. Okay. What is the purpose of this experiment? They have three way. A DNA can replicate in three way. There's a possibility. One is called conservative model. Conservative model. Second one is semi-conservative model. Semi-conservative model. Third one is dispersive mode of replication. Dispersive mode of replication. Okay, these are the three model of replication. Now let us see how did they do this experiment. What they did is Meselson stahl, they took E. coli colony, Escherichia coli. They took E. coli colonies. Okay, E. coli colonies. They grew these colonies, E. coli colonies, in a medium containing what? Medium containing heavy isotope. Which heavy isotope they used? They used isotope of nitrogen, that is 15N. They used this isotope, 15N isotope. This is a heavy isotope. They grew this E. coli in this isotope. After that, what they did is, after growing in the medium for few generations, they took few colonies. They took they took the colony from heavy isotope, from this medium, heavy isotope. Okay, after that they placed it in another medium. They placed these Escherichia coli from this medium containing heavy isotope, transferred to light isotope medium containing light isotope. Now they are going to do the experiment. Now what they did is, after that they are taking a sample from each generation. First sample they took, that is they called F0 generation. F0. What was the F0 generation? F0 generation is that they took E. coli sample from medium containing heavy isotope. That was just before placing into the light medium. After taking the sample from heavy isotope medium, they purified the DNA. They did, after purification, they purified DNA. After purification, they did density gradient centrifugation. Density gradient centrifugation. They did what? Density gradient centrifugation. Okay, they did density gradient centrifugation. What is the density gradient centrifugation? Density gradient centrifugation is that purified, I will tell you what is density gradient centrifugation. Purified sample of DNA, purified sample of DNA is centrifuged along with cesium chloride, to separate the DNA, into bands according to density. Purified sample of DNA is centrifuged along with the cesium chloride to separate the DNA into bands according to the density. This is what their centrifugation experiment. This is density gradient centrifugation. This is called what? Density gradient centrifugation. Okay. After centrifugation, what they found, I will tell you. They took a first sample that's called F0, that was just before placing into light medium. After centrifugation, in the spinning tube, they found one band which was in the lower portion. They found a band in the lower portion. This was heavy band. This is called which band? Heavy band. It was absolutely expected, heavy band. Why? Because when it was in the heavy medium, the DNA will take up heavy isotope. So automatically after centrifugation, you will get a heavy band because of N15. This was expected. After that, what they did is they took a sample from a light medium, that called F0. That is called what? That is called F1. F1 is what? They took a sample they took a heavy DNA, sample from heavy isotope, and they placed it in the light medium. So this was our DNA. This was heavy, correct? They placed it in the light medium. After placing it in the light medium, this DNA will replicate. So after the replication, they put it in the light medium, and they took a sample from the light medium now, and they did the density gradient centrifugation. What they found? They found a band. That was just in the middle. It was not in the top. It was not in the bottom. It was in the middle. It's like, you can say, intermediate. It's called intermediate. Neither light or heavy. It's intermediate band. Why it is intermediate band? To get intermediate band, there is a two possibility to get intermediate band. First possibility is that DNA is replicated. If, for example, I will tell you what are the possibility. There is a two possibility. One possibility is that when you placed heavy DNA into a light medium, one of the strand, that is H, will make a complementary strand, that is this, will have light band, light isotope or light DNA, light strand. It will make a light strand. So each strand will act as a template for a new strand, that is L. Each heavy strand acts as a template for a light strand. Okay, this is the one possibility to get intermediate band. This is like a hybrid. The another possibility is that you have a heavy strand, and when you place this heavy strand into a light medium, the another possibility is that a new DNA segment will be inserted like a patchwork. You have a tire, you can make a patchwork for the tire. A newly synthesized DNA strand with a light isotope will be intercalated between the heavy strand. So one will be H, L, H, L, like a patchwork. Same way, the other one. You have a heavy, the second heavy, and you have a patchwork. In these two cases, you get intermediate band. You don't have light band because you don't have a separate light DNA, neither you have heavy DNA strand. You have intermediate, you have a mixture of both, like a hybridization, like a hybrid. So intermediate band, this is called intermediate band. Now you want to know, now it's clear that you found after centrifugation in the middle, the band was in the middle. It means DNA is replicating either this way or this way. It is absolutely clear from this one, DNA is not replicating conservatively. Conservative means what? You have a double strand that was H. These both H are not used or not acting as a template for a newly synthesized strand, and it's not becoming a part of a new strand. Instead of going this parental strand with a new strand, two new strands are made. That new two strands are becoming complementary to each other. Parents are separate, the new strands are separate, the offspring is separate. There is no complementary between parental strand and a new strand. Complementary is between new strands itself. This is called as a conservative model. Then you will have heavy band separate and you will have a light band separate. Instead of having intermediate, you will find one heavy band here for the parental and for the light band here for the offspring. But you don't have that case here. You have just intermediate band. From this intermediate band, it is very clear that DNA replication is not conservative. DNA replication is not conservative. DNA replication is not conservative. Okay. Now you have a two possibility. Either it is semi-conservative or dispersive. Now we are going to check whether it is semi-conservative or dispersive. You took another sample that is called F2. What you did is that you are taking again the sample. How are you going to take next generation? You are going to do one more generation and you are going to check it. So let us see what was our previous experiment. Was, after doing the experiment, what they found, they took a sample from that is F2 generation. They did centrifugation. What they found? They found intermediate band. But they found light band as well. Previous experiment, they found only intermediate band. There was no light band. Now they are seeing both bands, light band as well as intermediate band. What does it mean? What it says? I will tell you what does it says. Now you find both bands, right? Okay, light and intermediate. Previously, intermediate was 100%, light was 0. Now intermediate band reduced to 50. Light band increased to 50. Let us see whether it is a 50 or no. Let us see now. Let us see. Okay, what we are going to do? You have a strand from the heavy. Okay, it was from the heavy medium. You place it in where? You place it in the light medium. So what you are going to find? You are going to find this way, right? 2 generation. This is the F0. Now I am going to make F1 generation. Let us make F1 first, then we'll make F2. So this is heavy. You have a complementary in the light medium, so that will be L. You have another stand, that is H, and the complementary light, both is which one? Intermediate band. That's what I said. You don't have a light band in the F1 generation. You have just a intermediate band, as I told you before. In the F1 you have just intermediate band. Now you are going to make from, remember that light and H, F1, you have a 2 stand. Now you are going to make a F2 generation. Okay, I will draw this picture again, then we'll make, this was our strand. Okay, now we are going to make F2. This is L. Okay, and this is another strand. This is H and this is L. Now you have this strand. I will take this H. What are you going to it? H. Okay, and now I will make another strand in the light medium, that will be L. Now you have this L. You have complementary strand L. Now I take this H, this one, same, right? H, you have L, you have this L, you have this L. So see, how many LH? 1, 2. Sorry, from this, how many LH? 2. How many LL? 50, 50. That's why I said 100% became 50 and 0 became 50. That's what I said. In the F2 generation, what did you find? You find light band in the, intermediate band. It was 100% in F1. It was 100% in F1, became 50. It reduced. Whereas light band, it was 0%. It has increased to 50. It means that the possibility to become dispersive model, is coming down, and the possibility to become semi-conservative is increasing. Why? If it was dispersive model, you will not find a separate light band. You will never find separate light band. You will always find hybrid, like a patchwork of LH. Correct? Dispersive model says that. So because dispersive model in the intermediate band says 2 things, but here when you do F2, you see that F2, when you do F2, intermediate band is coming down now, whereas the light band is increasing. Now to confirm that, they continued the experiment for F3 generation. You take a sample of F2 and make it F3. What was F2? I will tell you what was F2. F2 was this. You had H, you had H, you had L, and you have another H, you have another L, you had LL, correct? You had LL, correct? Now I will draw this one down, this one down, this one down, this one down. Okay, H you had, correct? This H, from this H you are putting in the light medium now, right? So it will be L. Here you have L. Complement will be L. Here also the same. H you had, you are in the light medium. L, you are in the light medium already, and you are L. So here also you will find L complementary to LL plus LL. Here also the same, LL L. Now see, 1, both are light band, 2, 3, 4, 5, 6. How many light band? Light band is 6. How many LH? 1, 2. Now light band is again increasing. So intermediate band is decreasing more. It means that the light band is becoming more stronger. Heavy band is intermediate band is becoming very weak. So it is very clear that intermediate band is becoming very thin, whereas the light band is becoming very thick. It is very clear that DNA replication is semi-conservative because dispersive replication possibility is coming very, very down. So it is very clear that DNA replication is semi-conservative mode of replication. Okay, this is the experiment. Okay, now let us discuss about that. Now let us see what is you mean by our what you will call, what are the importance of Meselson stahl experiment? What are the importance? Why do we need this experiment? This experiment is to know whether the DNA replication is conservative or semi-conservative or dispersive. So it is very clear that because it is semi-conservative mode of replication, it gives you an idea that DNA parental strand is used as a template. Each strand is used as a template for a new DNA synthesis. This is the points to be remembered. Importance: one is to know that whether DNA replication is which mode of replication. Second one is we can find out that one strand is parental strand is used as a template for a new strand. This is what the Meselson stahl experiment says. And because we proved that it is not a conservative mode of replication, what was clear that DNA replication is. So from this point we understood that DNA replication is not conservative. If it was conservative, the DNA new strand that was made, the new DNA will not be having a template from the parent. So our experiment proved that our DNA replication is semi-conservative. Thank you for listening. If you have any question, please comment. Thank you. Subscribe my channel.